Diabetic patients might benefit from an insulin-alternative protein. Insufficiencies in insulin synthesis may lead to diabetes, which is characterized by high blood sugar levels. A novel molecular route that controls blood glucose has been discovered by scientists at the Salk Institute and might lead to a new treatment for diabetes. Insulin's primary function is to direct cells to either consume or store extra glucose as blood glucose levels rise. Nonetheless, diabetes may develop if the body is unable or unwilling to make insulin. Of course, this implies that insulin is the primary treatment for diabetes, but there may be an alternative. An enzyme called FGF1 was found by the Salk researchers to serve the same job as insulin in controlling blood glucose levels a few years ago. Studies demonstrated that brain injections of FGF1 might successfully put diabetes into remission for weeks or months in diabetic rats. Researchers at Salk looked into the mechanism of FGF1 to see whether it operates in the same manner as insulin in the current study. Some of the functions of FGF1 are similar to insulin's, such as controlling glucose generation in the liver and preventing fat breakdown, or lipolysis. FGF1, on the other hand, was discovered to function through an entirely distinct chemical route. Insulin activates a signaling pathway that inhibits lipolysis by activating an enzyme called PDE3B. A variety of enzymes, including PDE3B, were tested on FGF1, and a different one, PDE4, was discovered to be used instead. A second loop with all the benefits of a parallel channel, says Jensa Sankar, the study's lead author and a professor at the University of California, Berkeley. Insulin signaling is disrupted in cases of insulin resistance. If one signaling cascade doesn't work, or another one may. So you can still manage lipolysis and blood glucose. This critical distinction may offer up new research avenues for diabetic therapy. An FGF1 modification might enhance PDE4 activity, or other sites in the system could be targeted, according to researchers. As co-senior author Michael Downs points out, FGF1's unique ability to produce prolonged glucose reduction in insulin-resistant diabetic mice represents a possible treatment path for diabetic people. We are hopeful that improved therapies for diabetes patients will result from our knowledge of this route.